Okay, so good morning, everyone. So thanks for the committee for inviting me for this symposium. It's my great pleasure to share our recent work with everyone. So my name is Chuan Wu. I'm the investigator from NCI. So my lab is interested in basically an immunology lab. We actually are interested in how neuronal reaction and immune reaction crosstalk control the barrier integrity, particularly on the mucosal surface. So what I'm going to talk about today is how the immune neuron transmission regulates gut sensation. So as we all know that the GI tract function not only as a major digestive organs, but it's also function as a sensory organ because there are so many different substrates go through the GI tract, like different bacterials, dietary fibers. So the intestinal epicidal as a barrier can not only absorb different nutrition, but also at the same time they perceive so many different signals from external stimulation. As I listed here, like microbiome signals, antigens, mechanical stimulations, and these signals and are very first contact with the intestinal epithelial barrier, and then they can be transduct or translate into different signals, including chemical signals and neuronal signals, which actually pass along through different nervous system into both local interneural network and then central nervous system to control both intestinal physiology and systemic physiology. So for a long period of time, it's been always a mystery that the intestinal epithelial, how they actually convert these external stimulations into chemical signals. So there's a brief introduction which published on Nature Review Neuroscience, showing that there are different actual receptors perceive these stimulations and then translate through complex machinery and then become neuropeptide or hormones and then pass along through different neuronal system. So our question obviously is, since we are more in immunology lab, so we actually are interested in how do immune response can participate in this process, in particular how they control gut sensation. So before I dive into our story, I just want to give also a brief introduction of the main role of this work. One of the main immune regulators, also a very old one, a IL-1 family member. So IL-1 family, as we all know, is probably the one of very first interleukin family being discovered in immunology. And then they all carry similar protein structures. But one of the members I'm going to introduce you today is the very recent one, IL-33. This one, this cytokine actually was discovered not more than 15 years ago and then become so popular in the field, both not only restricted in the immunology field, but also in the development in the metabolism, because this cytokine is particularly interesting as not only secreted cytokine, this is also a nuclear protein, which usually stay in the nucleus of the cells and then only be released, not in the conventional way, but only when the cell get damaged or have the tissue injury. And then this cytokine had been demonstrated play a lot of different roles in different biological process. At the same time, the receptor of the cytokine have two different subunits. One is its specific subunit, ST2, and the other common subunit, IL-1 receptor associated protein. So just want to give you a reminder that I'm going to talk a lot about ST2 as the specific receptor for the interleukin IL-33. So as one of the key components for the body defense machinery, the intestinal not only harbored a lot of commensal bacteria to maintain its homeostasis, but at the same time, during an evolution, the peristaltic movement for the gut also function as a very important defense machinery for the whole body, because it's actually facilitate the body to expel the toxin and then also facilitate the digestion process. So our indicator to study the gut sensation is cut in from this angle to understand the peristaltic movement, how the immune response function upon that. So to actually dive into the study, we actually do a very simple work. This is a daily administration of IL-33 IP injection for 1 week. And then we measure how fast the gut motility becomes. So you can clearly see here, after 1 week of administration of IL-33, the gut motility significantly improved. And this actually can be verified from both IL-33 knockout or the receptor ST2 total knockout mice. So these mice all show significant delayed gut motility compared with the wild type control. And then by using the in vitro assay, we have this organ-based system. We can actually take the gut tissue out of the mouse and then do the simultaneous autonomous contraction assay in vitro. So in this case, you can see every single peak represent a one-time contraction of the gut in vitro motility. And then you can see in this case, wild type has this rhythmic, very regular contraction. While if you compare with both IL-33 knockout or ST2 knockout, these mice actually show quite significant compromised gut motility compared with wild type. But the most interesting part is actually when you perform this experiment in the wild type case. So when during the tissue in vitro in the organ bath, as soon as you administrate IL-33 upon the tissue, you actually see an instantaneous increase of contraction of the tissue. And this actually can be seen on the IL-33 knockout tissue, but not in the ST2 tissue, which indicate that the IL-33 modulates the contraction or peristaltic movement of the gut tissue in a very rapid, instant manner, probably controlled by neuronal cells. So obviously, we want to ask the question, which cell type particular responsible for this IL-33 mediate gut motility. So I'm gonna not go through all the details. I'm gonna just jump right into the conclusion that we actually go through almost about 20 different specific cell subset. And in the end, we pinpoint that the epicidal cells, particularly if you delete the ST2, which is IL-33 receptor, upon the intestinal epicidal cells, then you can phenocopy the delayed gut motility as the total knockout, which obviously indicate that intestinal epicidal is the main responsible cells. And then in this case, you can even more clearly prove my point that if you delete ST2 on the intestinal epithelial cells, for example, you can see the very left two columns that wild type animal, when you apply IL-33, you can see increased gut motility. But if you do the same experiment on the conditional knockout, then if you abolish ST2 on the epithelial cells, whatever you do, IL-33 does not function anything upon these mice. So which indicate that epithelial cells really is the main cell type for this whole action. So now the question is, because this is rather a very quick response. So obviously we want to ask whether this IL-33 promote gut motility is through the interneuronal activation. So in order to answer this question, so we also carry out, or actually we design a new in vitro assay. So this is a piece of intestinal tissue. It's just to give you an idea of autonomy of intestinal layers. You can see there are 3 different structures in the intestinal tissues. But if you apply different reagents on top of these tissues, so what we do is we can use a long-distance confocal microscope to monitor the intelligent neurons, which particularly locate on the muscularis layer, which is also called myoenteric plexus. We can use GCAM indicators to monitor how the neuron responds upon in this particular structure. So I'm just gonna tell you that we actually using the specific intelligent neural markers PERT, and then using the transgenic animal model PERT GCAM animal. So what you can see here is under steady state, there's obviously some background on the intelligent neurons, but as soon as you apply allosurgery upon the tissue, then you see a neuronal, quite strong neuronal activation. And this is just the MCPG as a part control, showing that the whole assay worked properly. So with the quantification, you can see when you apply the whole tissue with the allosurgery, you can clearly see as soon as you put allosurgery on the tissue, there is a clear neuronal response, same as the palliative control. But the interesting thing is, as soon as you mechanically remove the epithelial cells, if I can remind you here, so because there are 3 different layers, on the very top mucosal layer actually are the epithelial stents. So we can, using mechanical force, to remove this particular layer. So as far as you remove this layer, what do you actually see? The result is whatever you put on allosurgery, there's no response whatsoever. The palliative control still has showed the response. So this actually clearly indicates that the epithelial cells is really responsible for allosurgery signal, and then to conduct, to pass along this signal to the interior neurons, control the motility. So the next question is, how does this happen? How do epithelial cells control this neuronal activation? Through the previous literature, we know that a lot of epithelial cells actually function as a chemosensor to perceive different signals, but most conventionally, these signals are come from mechanical stimulations or the microbiome metabolites. But there's no actually reports showing that immune signal actually can trigger this whole cascade. So after the chemosensor of the epithelial cells, particularly the enteroendocrine cells, receive these signals, they actually can start to function, release the hormones or chemopeptide, neuron peptide to conduct the further signal. So we collect all these informations and then form a panel. What we did is we inject allosurgery to the mice, and then within a very short period of time, we collect the serum and then use the serum to run the secretomics based on the collection candidates from the epithelial cells. So we actually got pretty lucky that only one particular substrate stand out, which is the serotonin. So we actually found that allosurgery stimulation can dramatically increase the serotonin production. And this actually can be proved by functional assay. So PPH1 actually is a serotonin synthesized enzyme. If we knock it out, that the entire peripheral serotonin was completely abolished. So similar readout is, you can see the very left 2 panels are Y-type animal with the allosurgery stimulation, the gut motility gets dramatically increased. But if you remove all the serotonin from the peripheral, then there's no response to allosurgery whatsoever, which actually indicate that the serotonin is the key mediators. So as I showed you that the serotonin is mainly actually synthesized from the intestinal epithelial cells, but one particular candidate or one particular cell type is called enterochromatin cells. They actually only occupy 1% of the total epithelial cell population, but they actually synthesize over 95% of the peripheral serotonin, quite potent cell subset. So what we actually try to address is whether this particular cell is responsible for the whole consequence. So first of all, we actually address that the serotonin CAGA, which is the enterochromatin cells marker, and then the receptor ST2 are co-localized. And then we genetically delete the ST2 on the specific enterochromatin cells. So you can see exactly the same phenotype that if you delete this on the specific cells, there's complete abolished whole.
About 15 minutes.
Okay, I'm almost done. And then also you can see accordingly that the serotonin level with the conditional knockout mice is dramatically reduced, and ALCL3 cannot rescue the whole phenotype. So with that, I'm not going to go to too much molecular details. I'm just going to summarize here what actually we believe happening is in the intestinal, the constant renewal of epithelial cells released ALCL3, which can be perceived or sensed by the enterocromping cells. The SE2 signal actually triggered the downstream PLC gamma signal, and that leads to the activation of the IP3R receptor, and then to stimulate the calcium inside of the cells to open the TRPA1 channel. These data I didn't have the time to show, but you can just look up to our paper. And this openness actually trigger further downstream cascade of vast calcium signal into the cells, which actually promote the serotonin release and then activate the interneuronal activity. And then consequently, we can monitor the gut motility and then further down to facilitate the parasites expulsion. So with that, I'm going to finish here. I'm going to thank my lab members, particularly Zoe and Jale, who are responsible for this study, and then a lot of collaborators I listed here, NIID, US Department of Agriculture, and NCI colleagues. And thank you for your attention.
Thank you, Dr. Wu. I have one question from the chat box from Yi Hong. Any evidence to support that interleukin-33 is released mainly by tissue damage as opposed to one of the unconventional protein sequencing pathway?
I'm sorry, I didn't get the question. What is the question?
Any evidence to support that interleukin-33 is released mainly by tissue damage as opposed to one of the...
I see. Okay, that's a great question. I won't be saying that it's only due to tissue damage, but I think it's dependent on the context. So in the intestinal GI tract, particularly, I think it's most because the rapid turnover of the epithelial cells. So ALCL3 is constantly released. The unconventional secreted pathway, yes, that's probably happening as well, but I don't think it's the main reason.
Okay, thank you.
I probably want to ask a question, Chuan, and so is this, I know serotonin in general is a very multifunctional neurotransmitter, and is this mechanism applied to other neurotransmitters? Such like dopamine, for example, and whether they are, so getting you some kind of, I think I'm not the neurologist, but I think getting you some kind of rewarding or sensing.
That's a great question. So first of all, I believe that's actually also happening in the central nervous system. I mean, the fun fact is serotonin, because the blood-brain barrier, they actually is hypophilic. So the peripheral serotonin cannot pass through the blood-brain barrier. So the serotonin in the central nervous system control the let's say behavior, let's say like depression. I will say more actually happening in the local, in the central nervous system strictly. But the release of ALCL3 probably different from the intestinal, because there's not so much renewal rate in the CNS. But they are, the ALCL3 basically is everywhere. So the oligodendrocyte cells has the ALCL3, so they will release the ALCL3 during the, let's say, neural repair. For example, in the stroke case, in the stroke, these cells are dead, and then they will release ALCL3. Yes, I believe that this only happen in the enterochromium cells, but on the neurons, they probably also will happen. But I'm not sure if they go through the same machinery. Yeah, so we found this machinery through the PLC gamma, but not necessarily in the neuron. But I believe that the neuron can release the neuron peptide through the immune signal input.
Thank you. Given the time, although there's still 2 questions in the chat box, I will ask them to ask you later, Dr. Wu,
Sure.
in the, yes, thank you. And welcome our next speaker.